Hello students, welcome to class PM. We are discussing acids, bases and salts. In the previous class we learned acids and bases and their properties, and some examples also we have seen for both of them. In today's class we are going to discuss about how to identify acids and bases. For example, in a beaker, in a watch glass, I have given you one unknown substance. I did not tell any name and I have given and asked you to identify whether it is an acid or a base or a salt or neutral. So how do you identify? You can say that we know that the substances which are sour in taste are acids and which are bitter in taste and slippery to touch are bases, and you don't try to touch it because I already mentioned acids. Some of the acids are corrosive, they are destructible, they are dangerous. So if you touch, it may burn your skin. If you taste, it will damage your tongue. So it is not correct way of identifying a substance is the acid or base because they may be dangerous, they cause harm to us. So there are special things or special arrangement is there to identify the given substance is the acid or a base or it is a salt. So those special arrangement is indicators. What these indicators will do? Indicators are the substances, they will change their color from when it is added to acid or bases or neutrals. Okay. When a particular indicator is added to acids, it will get a one color. If it is added to bases, it will get another color. Okay. By changing their natural color, their characteristic color, so they will identify the given substance is acid or bases. So what are the indicators? Indicators are the substances will useful to identify the given substance is acid or a base or neutral by changing their color. Okay, so there are two types of indicators again, the natural indicators and synthetic indicators. We will first discuss what are the natural indicators. Natural indicators. So first natural indicator we will discuss about is litmus. So what is litmus? Litmus is a extract which is extracted from a plant called one kind of plant sources that is called lichens. So what are these lichens? Lichens are the group of algae and fungi together. Okay, when algae and fungi lives together, it forms the lichens. Okay, algae means, so in the rainy season, if you observe the top of the roof or on the staircases or on the tanks, green color layer will be formed. That is called algae. A small green color layer will be formed. That is called algae. And what is fungi? If you keep bread for many days and it gets spoiled by getting into fungus. Okay, it is attacked by fungus. If you observe that fungi attacked bread mold, so you will see that there is a green color molds. You will see that some different color molds like this. You will see that those are called fungi. If algae and fungi lives together, it is called lichens. It's called lichens. So this litmus is extracted from the lichens. Litmus is extracted from a plant source called lichens, and this litmus is a solution or it may be available in the form of paper strip. Okay, the litmus, which is the natural indicator, is in two different forms. That is, it can be available in the form of a solution. If the solution is blocked, you just pour it on a filter paper and dry it and it will become a litmus paper. So it is also available in the paper stripe. Okay, now these paper stripes, mostly we will use the paper stripes to identify the bases and acids. Two colors of litmus paper is available, that is blue litmus paper, red litmus paper. Blue litmus paper and red litmus paper. So how it will indicate the given substance is a acid or base? For example, in a beaker I have taken one unknown solution. I don't know what is this. It is an acid or a base, I don't know. It is some X substance I have taken, I don't know. So I have two indicators with me, that is blue litmus paper and red litmus paper. So I kept this blue litmus paper in it. I kept the blue litmus paper stripe in the solution. So immediately it changes to red color. Blue litmus paper is changed into red color. So then I can say that the substance X is a acid. Okay? If a blue litmus paper changes to red color, then the substance which changed the blue litmus to red color is called acid. Next, I have taken another stripe and I have taken another solution. So in another beaker I have taken another solution, that is a solution Y I have taken. In this I kept this red litmus paper, and this red litmus paper changed into blue color. So red litmus paper, when I kept in this second solution, the Y solution, it changes into blue color. So I can conclude that the solution in the Y beaker is a base. So from these two experiments, what I can conclude is the substance in which blue litmus changes to red color is acid. The substance in which red litmus changes to blue color is a base. The simple way you can tell it, or you can conclude that like that: blue litmus changes to red in the presence of acids. Blue litmus changes to red color in acids. Red litmus changes to blue color in bases. This is the way you can conclude the whole topic. Okay. Blue litmus paper changes its color to red in the presence of acids. Red litmus paper changes its color to blue in the presence of base. If you put it in the neutrals, no change in the color. Blue litmus paper remains the blue and red litmus paper remains the red. And if I put the red litmus paper in the acid, what happens? No change in the color. Only the acid will change blue to red, but no change in the red litmus paper. The same way, base also, if I take a blue litmus paper, I put it in the base, there is no change in the color of blue litmus paper because it is a base. Base does not show any change for the blue litmus paper. Now we will take some examples now. We will try to understand more better about the litmus paper. Now we will do one experiment. With this experiment, we will try to understand more depthly about the indicators, especially litmus. So for this experiment, we will take the three samples. In one beaker, I will take the lime water. In another beaker, I will take the lemon juice. In third beaker, I will take the salt solution or sugar solution. Anything we will take. Now I will take some stripes of red litmus, some stripes of blue litmus. Now first I am taking, what I am doing, I am taking a blue litmus and I put that stripe in the lime water. Okay, I took the blue litmus and I put it in the lime water. Then what I find, there is no change in the color of blue litmus. No change in color of blue litmus. Next, I have taken the red litmus and I kept in the lime water. Then immediately that red litmus paper changed into blue color. So we know that bases will turn red litmus to blue color. So that I can conclude this lime water is a base. Now I have taken my second sample, that is lemon juice. So lemon juice, I have taken first blue litmus paper, I put it in the lemon water. So immediately that blue litmus paper changed into red color. So it's changed into red color. Next, what I have taken, again I have taken the red litmus paper and I kept in the lemon juice. So there is no change in the color. No change. Red color remains same. So we know that acids turns the blue litmus to red, so that I can, since the lemon juice turned the blue litmus to red, then I can conclude that the substance with me is acid. Next, I have taken the again blue litmus paper, I kept in the beaker of the third solution, that is salt water. Then I found the blue litmus did not change its color, so no change in color. Then I have taken the red litmus paper, again I have kept in the salt water's beaker, so there is no change in the color of red litmus paper. So red litmus paper color is not changed and blue litmus paper color also not changed. Then I can conclude that the substance with me is a salt. And above all, I can conclude that red litmus changes to blue in the by bases. Blue litmus changes to red by acids. This is the way it works to identify the given substance is acid, base or salt. So in the next class we will discuss about some other natural indicators apart from litmus paper.